Sterile Processing Department is responsible for cleaning, sterilizing, and storing reusable tools and equipment in a hospital. It is easy to understand how tools and various pieces of equipment can become both dirty and contaminated during surgical and other medical procedures. Central Supply ensures that the contamination and illness-causing germs are eliminated before the tools are reused. Germs is a simple term that applies to those microscopic particles that can cause illness or infection. We tell children to wash their hands to kill germs so they don't get sick. Sterile processing does this same thing in a very big way. Germs can be classified into groups of pathogens. Remember, the word pathogen means anything that can cause illness. This can be a disease or an infection. The 5 classes of pathogens that we'll look at are bacteria, viruses, protozoa, fungi, and prions. We'll take a look at each one. Bacteria. Bacteria are generally microscopic, one-celled organisms that do not have a nucleus. Remember, the nucleus is essentially the brain of a cell. The DNA is inside the nucleus, which is a blueprint for the cell to tell it who it is and what it is to do. The DNA for a bacteria are not enclosed inside the nucleus. It is simply inside the cytoplasm of the cell. Bacteria come in 3 common shapes: spiral, like a corkscrew, rod-shaped, and round. A group of these looks like a cluster of grapes, and each shape has a specific name. The spiral is spirella, the rod-shaped is bacilli, and round is cocci. Some types of bacteria have a tail called a flagella that helps them move through liquids. An infection can be caused by just a few of these microscopic bacteria. In the right environment for growth, the bacteria can reproduce, and very quickly there can be so many that an illness is caused. Bacteria reproduce by a process called binary fission. The blueprint for the cell, which we call the DNA, makes a copy of itself, and then the copies separate from one another. The cell divides into 2 new cells, each with a complete copy of the DNA. This can happen very quickly. There are millions of bacterial cells on and in a normal human being. Most are beneficial and help with processes such as digestion. Some are neither helpful nor harmful, and some have the ability to cause disease. The most common diseases caused by bacteria are cholera, syphilis, leprosy, bubonic plague, tuberculosis, and some types of pneumonia. There are many ways to classify bacteria. One way is by whether or not they require oxygen to live. Those that do are called aerobic. Bacteria that grow and reproduce where there is no oxygen are called anaerobic. The prefix "an" means not or without, and aerobe means requires oxygen. So you can see that anaerobic means not requiring oxygen. Anaerobic bacteria can cause nasty infections inside human body tissues where there is no oxygen. Aerobic bacteria are more likely to cause infections of the skin or other body parts that are exposed to air. Bacteria can also be classified by the conditions in which they grow. Psychrophiles like cold temperatures. Phile is a suffix which means likes. A chocophile might be someone who loves chocolate. A bibliophile likes books. Psychrophiles grow best at cooler temperatures around 60 to 70 degrees Fahrenheit. Mesophiles like moderate temperatures. Meso is a prefix that means middle or intermediate. They grow and reproduce in temperatures from about 70 degrees Fahrenheit to 115 degrees Fahrenheit. Since the human body is 98.6 degrees, mesophils are bacteria that can live in and on them. Thermophiles prefer warmer temperatures from 120 to 160 degrees Fahrenheit. Therm is the same prefix used in thermometer, and it means hot or heat. Thermophiles do not survive in or on the human body. Finally, bacteria can be classified by how they are identified in the lab. Two tests involving stains are used for testing bacteria in the lab. Number one is the Gram stain, and it's used to color bacteria on a microscope slide. Those that stain purple are called Gram-positive, and those that stain red are Gram-negative. This test is used as a first step in identifying the type of bacteria that may be causing an infection. These are the most common Gram-positive and Gram-negative disease-causing bacteria. The Ziehl-Neelsen stain is used to identify bacteria that are known as acid-fast. This is the second type of classification according to stains. They retain a red color when tested with the Ziehl-Neelsen stain. The most common disease-causing acid-fast bacteria are Mycobacterium tuberculosis that caused, you guessed it, tuberculosis, and Mycobacterium leprae that causes leprosy. Bacterial infections are treated with antibiotics. However, there are some bacteria that have become resistant to antibiotics, and those infections cannot be treated very well. MRSA, or MRSA, is the shorthand name for methicillin-resistant Staphylococcus aureus. This is a nasty skin infection. The other is VRE, or vancomycin-resistant Enterococcus. Both of these are very serious infections and can become a problem in hospitals. We've learned quite a lot about bacteria, one of the primary pathogens we are concerned about in the sterile processing department. If you think it will help you, click on the link for the guided notes for bacteria, print out a worksheet, and watch this tutorial again, filling in the blanks. In this way, you can more easily learn the material you'll need to know for the certification exam.